Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video 2.85, we shall look or discuss eicosanoids. So we'll have an overview of eicosanoids. So eicosanoids are lipids that are made from 20 carbon fatty acids such as EPA and arachidonic acid. So just to remind you, if you look at the structure of arachidonic acid, there's a carboxyl group and the rest is basically an alkyl chain. Okay, so basically an alkyl chain. And then you can see the double bonds here. So that's how we will represent arachidonic acid as a cartoon in these videos. Now, when you look at the eicosanoids, such as prostaglandins, prostacyclins, thromboxanes, leukotrienes, lipoxins, you see the carboxyl group, which is here. You see the carboxyl group, so that's common because these are made from these fatty acids, so which have the carboxyl group. But what you see is the changes in the alkyl chain. You see the formation of sometimes cyclic structures like this, cyclic structures. You see oxygens. You see oxygens, hydroxyl groups, oxygens. Okay, so you see a lot of oxygens in the alkyl part of the fatty acid, and this is because these the way these chemicals are synthesized, eicosanoids are synthesized. Oh, by the way, eicosa in Greek means 20. Is because they use these enzymes called oxygenases. These oxygenases use oxygen as a substrate, so they basically add oxygen into this alkyl part of the fatty acid. Okay, these enzymes, these oxygenase enzymes, two very important, cyclooxygenases, which we'll simply abbreviate as COX or COX, lipooxygenase, which we'll abbreviate as LO. So basically, the eicosanoids are compounds where oxygen is added in this part of the fatty acids, such as your EPA and your AA. As healthcare professionals, we really don't need to go into the details of the chemistry of eicosanoids. Basically, the chemistry is very complex and is not much use in going through details of the chemistry, but it's good to understand some of the nomenclature, some of the definitions, so that when you read a medical article or you read about how a drug works, it's good to know a little bit about the way these eicosanoids are named. So a little bit about prostaglandins first. So all these prostaglandins have this 5-membered ring. 5-membered ring. So you see that in all the prostaglandins. And you can see that these are all 20 carbons. That's why they're called eicosanoids, carbon 1 all the way to 20. And the different types of prostaglandins, so there are prostaglandins A, B, C, D, E, F, and so on till class K. And they all have different substituents. So you can see different substituents. Prostaglandin A has this substituent. Prostaglandin E has this substituent. Okay, so the substituents on the rings differ from prostaglandin A, B, C, and so on. Okay, so all the classes A to K have different substituents. So besides the classes A, B, C, and so on, sometimes you see numbers. Like here is prostaglandin E1, here is prostaglandin E2. So you see the same substituents on E1 and E2. You see the ring, but the difference is E1 has only one double bond, while E2 has two double bonds. So the numbers here denote the number of double bonds. Then the chemistry can get a little bit more complex. Sometimes you see things like alpha, beta, and that's because at carbon 9, if there is a OH present, then this alpha or beta basically denotes the stereochemistry at this carbon number 9. So for healthcare professionals, once again, it's really not that important to get into the details. We won't spend any time on talking about things like stereochemistry for the eicosanoids. But just remember prostaglandins, different classes, and then the number of double bonds. Prostacyclin is PGI, class I, and it's kind of a unique prostaglandin because this is C6, 7, 8, and 9. And between C6 and C9, you can see there's an oxygen connecting the two. So that gives this prostacyclin or PGI two 5-membered rings, 1, 2 5-membered rings. And this ring again is formed because there's an oxygen bridge connecting 6 and 9, which you don't see in the other classes of prostaglandins. Thromboxane is another type of eicosanoid. Unlike a prostaglandin that has a 5-membered ring, thromboxane, the ring is 6-membered and heterocyclic. Heterocyclic simply means in the ring. Like here is only carbon, 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 carbon. That's not heterocyclic. But if the ring has carbon and any other atom, in this case oxygen, that makes it a heterocyclic ring. So thromboxanes, basically you can see this 6-membered heterocyclic ring. Another class of eicosanoids are called leukotrienes. Leuko from leukocytes or white blood cells. Trienes mean 3 conjugated double bonds. So you can see there's a double, then there's a single, there's double, there's single, there's double. These are called conjugated. In organic chemistry, we call these conjugated double bonds. Okay, so 3 conjugated double bonds, leukotrienes. Another class of eicosanoids are called the lipoxyns. Now these are trihydroxy, 3 hydroxyl groups, trihydroxy, tetraene, conjugated tetraene, so double, single, double, single, double, single, double. So there are tetraene, 4 conjugated double bonds, tetraene. So lipoxyns are trihydroxy conjugated tetraene compounds. Fancy name for these very, very important compounds, lipoxyn. So what are the functions of eicosanoids? Basically, functions can be divided into 2 types. One is their functions in normal physiology. First, let's look at their functions in normal physiology, and then we'll look at their functions in a very important process called inflammation. Okay, so let's look at a few examples. Again, there are many eicosanoids. This field is very complex. So let's look at this sort of an overview. So protection of the stomach is, for example, if you look at prostaglandin E2, it is known that one of the properties of prostaglandin E2, or one of the functions, is that it protects the stomach. So when this prostaglandin E2 is released by a cell, then it will go bind a receptor on a nearby cell. These receptors for E2, there are 4 types called EP1, EP2, EP3, and EP4. Now it's important to note that these eicosanoids are local signals. That means they only work nearby the cell that secreted them. Okay, so they work only nearby to the cell, to the microenvironment where they are secreted. That's because they get broken down very fast. Okay, so first of all, they're not very stable. Secondly, they get broken down by all kinds of enzymes very, very quickly. So these eicosanoids, they are secreted, they do their job, and they are quickly destroyed, very quickly destroyed. So how does prostaglandin E2 protect the stomach? One is it inhibits gastric contraction. The more the stomach contracts, the lower the volume, which means the more concentrated the acid can become. Remember, the stomach secretes acid. The stomach is normally protected by mucus, so the destruction of the stomach mucosa by the acid is prevented partly by the mucus production. And one of the things that prostaglandin E2 does, it actually increases the mucin protein production. Mucus has a lot of proteins called mucin, so by increasing protein synthesis, the mucus cells produce more mucus. Okay, in this case, the prostaglandin E2 binds receptors EP3 or EP4. Another thing that this prostaglandin does is that we know that irritants, including hydrochloric acid and even ethanol, so some irritants can actually make these mucus cells undergo apoptosis. So these mucus cells programmed to commit suicide or self-destruct is initiated by these irritants. So we know now that this prostaglandin E2 actually stops this program from happening. So decreases the irritant-induced apoptosis of mucus cells. And here the EP2, EP4 receptor is what is bound by the prostaglandin E2. So other ways by which the stomach is protected is increased bicarbonate secretion by the duodenum. Prostaglandin causes more bicarbonate, and of course bicarbonate helps neutralize the acid. And importantly, it also decreases acid production by the parietal cells. So the production of HCl by parietal cells is decreased through the EP3 receptor. So again, these are signals. So when the signal binds the receptor, it leads to cell signaling that then affects all kinds of biological activities in various cells. Another normal physiological effect of prostaglandins, again, let's go back to E2, is the influence on blood vessel dimension. So most commonly, E2 binds to EP2 or EP4 receptors on smooth muscles found in the blood vessels, and they relax the smooth muscles, and that makes the blood vessel bigger. Okay, vasodilation. Sometimes there is vasoconstriction, rarely. That occurs through EP1, EP3. This vasodilation effect is very quick, occurs in about 20 seconds after you infuse the prostaglandin. But as I mentioned, these prostaglandins are very short-lived, so they are destroyed very quickly. So the effect lasts only about a few minutes. So in Blood vessels, smooth muscles of blood vessels, the most common effect that we see for E2 is muscle relaxation, and that leads to vasodilation. But in other muscles, such as, let's say, the uterus smooth muscle, the effect is opposite. There is a muscle contraction, causes the uterus to contract, and that occurs when the prostaglandin binds these receptors, EP1, EP3, in the uterus. So this shows that the same prostaglandin can cause one effect in one organ and another effect, opposite effect in a separate organ. Another normal physiological effect of prostaglandins is an example of prostacyclin, PGI, prostacyclin that binds the IP prostacyclin receptor, the IP receptor. Here, for example, you see endothelial cells produce prostacyclin, which binds to the IP receptor on smooth muscle cells, relaxes the muscles, and the blood vessel increases in dimension, vasodilation. A very important effect of endothelial cells is also production of prostacyclin, which prevents platelets from aggregating. And that's very important because under normal circumstances, we don't want platelets to aggregate. So one of the functions of endothelial cells is the production of prostacyclin that binds to the platelets and prevent the modulation of their actin cytoskeleton, which can cause platelet aggregation. So under normal physiology, that's one reason why platelets do not aggregate, is because of this prostacyclin production by the endothelial cells. So besides having a role in normal physiology, perhaps the most important aspect of eicosanoids is their role in inflammation. And of course, in the future, we will have videos on the biochemistry of inflammation. So this is how the body responds either to some sort of injury to the body or to the presence of some foreign substances such as microbes. So inflammation is a very, very complex and extremely important process that happens in the body, really to protect us. But in the process of protecting us, it might also hurt us. So in this slide, you can see partly the how these different eicosanoids are synthesized. Just an overview. So let's say there is a tissue injury or some sort of stimulation, mechanical or chemical. Many cells, remember these arachidonic acids are in the cell membranes. So that stimulation can cause this enzyme phospholipase A2 to break off that arachidonic acid, which is part of the cell membrane, part of the amphiphilic lipid. So that releases the arachidonic acid, okay, through the breakage by phospholipase A2. And once arachidonic acid is released into the cell from the membrane, then various enzymes such as lipoxygenase as well as cyclooxygenase can convert the arachidonic acid into various eicosanoids such as leukotrienes here or prostaglandins here, various prostaglandins with the help of prostaglandin synthase, prostacyclins with the help of PGI synthase, thromboxanes with the help of thromboxane synthase. So basically from arachidonic acid, we can get many different types of the eicosanoids. So these eicosanoids are produced by many different cell types, including a variety of your immune cells, mast cells, endothelial cells, as well as various epithelial cells. So a lot of cells are able to produce these eicosanoids. So for example, if there is some infection, some bacteria that has entered the tissue that triggers inflammation, so then we have eicosanoids such as thromboxane, which go to the smooth muscle and causes contraction, and that causes vasoconstriction. So this vasoconstriction makes a blood vessel smaller, and it also helps in that case by making a blood vessel smaller, and by having direct effects on platelets, it causes the platelets to aggregate. So basically forming a sort of a prison around this infected area to make sure that those whatever those microbes are not going to escape out of this inflammatory site. And then the other type of eicosanoids, such as your leukotrienes, they can cause effects such as increase the vascular permeability, the space between the endothelial cells, and many of these leukotrienes are very, very good chemoattractants. So these leukotrienes bind to leukotriene receptors, the BLTR receptors on various cells, neutrophils, macrophages, eosinophils, and that brings to cells. Remember, these cells are traveling in your bloodstream. So now this chemoattractant, this leukotriene concentration is higher around here, and that is going to cause these cells to start migrating towards where the bacteria, where the microbe is, where the infection is happening. Prostaglandins are also known to cause fever. For example, PGE2 binds the EB3 receptor in the hypothalamus, and that causes the set point for your thermostat, for your body temperature. The set point is increased, and that causes the sensation of fever and chills. Prostaglandins can also bind nerve terminals, these nerve terminals known as nociceptors, terminals of sensory neurons. And when they bind, they trigger an action potential that then goes to the brain and gives us the sensation of pain. So hopefully with these effects of all these various prostaglandins and leukotrienes and thromboxanes, these microbes will be eaten up by macrophages, neutrophils, and so on, and eventually be eliminated. So this inflammatory process has a lot of side effects too. The increased vascular permeability can lead to fluids escaping out of your nose and eyes and so on. Fever that can get pretty high, and there is pain. During allergies, there could be very serious consequences due to bronchoconstriction. So clearly these, when this inflammation goes out of control or becomes too extreme, leading to serious signs and symptoms, we need to control it. We need to decrease the inflammation. Therefore, there are a lot of, not surprisingly, a lot of anti-inflammatory drugs. The non-steroidal anti-inflammatory drugs such as aspirin, ibuprofen, and so on, they basically affect these steps here, the cyclooxygenase. While the steroids, because steroids go right to the genes, to the DNA, they can affect many different steps of inflammation, including stopping the production of cyclooxygenase, as well as decreasing the production of phospholipase A2. So thereby decreasing the amount of eicosanoids that are causing these signs and symptoms such as bronchoconstriction, fever, pain, secretions, and so on. So during inflammation, there could be a lot of damage to endothelial cells or epithelial cells. So once the offending, the microbe, for example, is destroyed, is totally removed, then we have to go back to the normal, homeostasis. The tissue has to be brought back to normal. And that is the second part of the inflammation, which we call resolution of inflammation. So many chemicals are involved in the resolution of inflammation. One that we'll talk about here is this eicosanoid called lipoxins. You can see how they are synthesized. Epithelial cells, again by breaking phospholipase A2, getting arachidonic acid, and then lipogenase enzymes like 15-LO convert that into intermediates, which then can be converted into lipoxins. Similarly, the neutrophils and macrophages can make their own lipoxins. It's interesting to note that aspirin can actually modify the COX-2 enzyme and give you intermediates that can be made into lipoxin derivatives, which are equally effective, which are also very effective in their function. So aspirin, low dose aspirin has a benefit because it allows this formation of, or encourages the formation of special lipoxin molecules. Lipoxins can also be converted or can be made from leukotrienes. So 5-LO makes the leukotriene, and then this leukotriene can be converted into lipoxins. And these leukotrienes, as the inflammation is slowing down, the problem has been resolved, the bacteria have been killed, then these leukotrienes can be taken up by other cells such as platelets, and platelets have their own LO enzymes that can convert these leukotrienes into lipoxins. So this synthesis of lipoxin from leukotriene, that the leukotriene is made by one cell and then the lipoxin is made by another cell. This is called transcellular biosynthesis. So you can see lipoxins can be made by different cells and different biosynthetic routes. And also can see that aspirin has a beneficial role in lipoxin synthesis. Okay, so why are these lipoxins useful in resolving inflammation? Once inflammation is done, then all the excess neutrophils that have been made, they have to be eliminated. And if they're activated neutrophils, we must stop their activation. Otherwise, the inflammation will continue. So we don't want continuous accumulation and activation of neutrophils, which can destroy normal tissue. So we don't want that. Then the second thing is the early macrophages help in inflammation, but then the later macrophages, when the inflammation is going down, those other macrophages they change and they start helping in the process of resolution. They clean up dead tissue so that you can remake your tissue. Okay, so these lipoxins, they actually delay the apoptosis of these beneficial macrophages so that these macrophages can go on and help in resolving the inflammation. Lipoxins are also known to inhibit excess antibody. Once the inflammation is done, we don't want chemicals that promote inflammation, such as antibodies and pro-inflammatory cytokines. We want their production to stop so that the inflammation is resolved, stops. Because we know now if inflammation is not stopped, that leads to chronic inflammation, and chronic inflammation is known to be a cause for many, many diseases in humans. Many diseases. So it's not surprising that today a lot of pharmaceutical companies and scientists are studying these lipoxins very carefully so that we can use them to stop chronic inflammation that has been associated with many human diseases. Okay, so that is an overview of eicosanoids. Again, please don't forget to subscribe and we will see you in the next video.